everyone. So thanks for the invitation and give me this opportunity to talk about some of the work we do in my group in Toulouse. Yeah, so I work for INRAE and I'm in the animal genetics division. And my lab is so situated in the south of France. So as part of the animal genetics division, the organisms I work with usually in terms of data, when I work on data, are livestock species and different kinds. And this is just to give you a kind of a context of what we're interested in. And so where do they come from and what is their evolution, because this is what we study. So most main livestock species have been domesticated somewhere about 10,000 years ago. Then lots of things happened, which we don't know exactly what, but people are investigating that for thousands of years. They've followed humans through the spread of agriculture. So in Europe here, but of course also in Asia, in Africa, and then later in the Americas. And there's been probably a lot of local adaptation, hybridizations between populations, and also hybridization with their wild relatives. And then a few hundred years ago started actual modern genetic breeding with the use of statistics and now the use of genomics. And genomics is now used routinely in livestock breeding. So you can maybe see this trend of net merit for Holstein cattle population. You can see there's quite of a steep increase in the genetic merit around 2010, and that's when they started using genomics for selection. Something that's not shown here is that the inbreeding has also increased quite a lot. So there's also a loss of genetic diversity that's accompanied with this gain in genetic progress. So through all this history, what we have today is actually very wide genetic diversity in this species. So what you have on the right, it's a bit small, but it doesn't really matter. It's a sample of goat population that has been sampled through most of the world. And yet we are missing a large part of Asia in this project, but you have Europe and Africa. And all these little tips on the trees are different breeds, different populations that are bred throughout the world. And they all originate from one population of Capra aegagrus in what is now Iran and Syria and Anatolia. So, and they spread out. And so what we do is we have this contemporary data on all these different breeds. So not always all of this. And we try to figure out what happened in the past. So the questions we ask are both contribute to some fundamental questions and also to applied questions and applied projects. So in terms of demography, what we want to do is to reconstruct the history and the genetic history of these populations from contemporary data. In my group, we don't work with ancient DNA, but there are other groups in the world who are also using ancient DNA to answer this question. And that helps understand the history of agriculture. And in terms of application, that also helps to know what is the genetic diversity, characterize the genetic diversity, and try to conserve it as much as we can, because it's very important. In terms of selection, we've been working a lot on trying to find the signatures of selection on the genome of this species. So identify the main genome regions that are involved in selective process that have been going on since domestication. And that helps to understand the biology of adaptation, of course. And from an application standpoint, it's also good information about the potential functional importance of mutations, because if you can find mutations that have been under selection, there's probably something functional about them. And so that also helps in the annotation of genomes. And more recently, we've been working on polygenic selection. So livestock population, at least in those populations that are genetically managed and selected, we can think of them as long-term ongoing artificial selection experiments for years and years. So they studying these populations can help to understand how phenotypic selection and genetic selection shapes genetic diversity and how the genome is responding to selection on phenotypes. And all the phenotypes that are selected in these populations are complex traits, quantitative traits. And also modeling this phenotypic polygenic selection can improve the prediction of complex traits and possibly the efficiency of artificial selection. So that's things we do. So I've said our trade in my group is to evaluate and develop new methods to analyze data, statistical method, and improve inference on these different subjects. Today, I'm not going to talk about all of this. Don't worry, I'm just going to focus on this last question, which is something we've been doing recently. Okay, so the objective of this talk and this work is to try to contribute to a better understanding of the genetic architecture of complex traits. What I mean by genetic architecture is how many variants there are that affect a trait, what is the distribution of their effects on the trait, and what is the contribution globally of these effects to phenotypic variance. And so that would be kind of a stable state question, but we also want to understand how does this genetic architecture evolves over time, and in particular when we are selecting individuals. Or I said we because I work on those livestock, but any kind of selection happens. Okay. So you can ask this different question in different contexts, and today I learned a lot about what you do on this question on experimental evolution. So my context is artificial selection. And also people working in natural population also have these questions, and sometimes the objectives are a bit different in terms of, so we are interested in predicting the effect of selection, improve response to selection, and manage the genetic diversity in the long term in those populations, whereas you want to understand the biology of population adaptation to new environments. And maybe in natural population, the question is, how do you find the traits that contribute to fitness? Whereas in livestock, of course, we know which traits contribute to fitness because they are designed by men. Okay, so when we want to study the genetic architecture of complex traits, there are different ways. So of course, you can do QTL mapping or GWAS. So that means your question is, how does genotype affect my phenotype? So your phenotype, Y, is the response, and your conditioning on the genetic diversity, you have G, and you want to find out what are the QTL effects, betas. And of course, when you do this for a particular locus, then you want to account for the fact that you have other QTLs on the genome, and so you have usually a polygenic effect in there. And of course, some non-genetic factors. So that's a typical, and what I represented here is just a distribution of the trait in gray, and then you have the 3 possible genotypes at a QTL that differ in the mean, and then you have variance across that. Okay, now if your trait affects your fitness, then what happens is that from one generation to the next, only certain individuals are going to reproduce. And when you mate those guys on the right, next generation after, then you get a different distribution of your genotypes. So what that means is that when you have fitness traits, when you're looking at fitness traits, the phenotype will affect the genotype over time. And so then your question is a bit reversed because your response is the genotype. And what is a covariate that affects your phenotype, your genotype, is a phenotype and some selection coefficient, some effect of selection. But when you do this kind of analysis, you want to account for the fact that there are also other factors than selection that affects your phenotype, and that are genetic drift, mutation, migration. So you need a model for the neutral evolution of your genotypes, which can be quite complicated if your population evolves and your individuals mate in complex ways. But what's sure, and what we want to leverage when you're studying genetic architecture, and what I will talk about in this talk, is that selection should leave genomic signatures at QTLs that we can find. So when we're looking at genetic architecture, we can look at it both ways. How does my genotype affect my phenotype? And how do my selection on my phenotype actually leaves traces on my genome. And today I will talk about how we can put these two ideas together. Okay, so if we think about what would be the ideal setup for doing this, so we want to have a population that evolves for selection on a known trait. And ideally, we want to have genetic data on everyone and maybe over time to study this effect of selection over multiple generations. We want to have individual phenotypes, so we can do a GWAS, for example. If we can know the demography, it's better because this, nuisance R effect term in these previous equations is known. And ideally, large population and sample size with replicates. Okay, so you can't have all of this, but we do have some of that. So I would say almost all except the last one. We don't have, in the work I'm going to show you, populations, or they can be large in terms of number of animals, but in numbers of reproducing animals, they tend to be small. So small effective population size. And okay. So the breeding programs are actually good models to study this question. And I will show you what we are doing. Okay. So in this talk, I will talk about two projects we have currently in my group. So one is the quantitative estimation of selection coefficient from genetic time series. And this is work we've been working in the past, and we are still continuing working on that with a PhD student called Paul Bunel, and with colleagues, in particular Simon Boitard. And the second part of the talk would be about how do we model the joint distribution of selective and quantitative effects, which is more of the title of the talk. But I wanted to talk to you about Paul's work because it's important to understand what's going on. In the second part, which is a work that was performed by Elise Tourette, who was a postdoc with me, just recently took a position in RAE 2, but in Paris. Okay. So estimating selection from genetic time series. Okay, so, and many people here are familiar with this kind of data. The idea is that we want to study, to model the evolution of the allele frequency in a population over time. But we don't have perfect information. So we don't know exactly what is the allele frequency in the population, and not at every generation. What we do is we sample alleles from the population every so often, sometimes regularly, sometimes. So in our case, we could have that every generation, but there are other contexts where you only have every 10 generation, every 5, whatever. So the question is, from data Y, can I reconstruct the trajectory in gray, and can I affect this trajectory with a selection coefficient? Can I find what is the most likely selection coefficient that leads to this trajectory? Okay, so if we think about how genetic parameters change these frequency trajectories, if you have, so the genetic drift that affects the random change of allele frequency from one generation to another, the smaller the population size, the higher the variance. So drifts only affect the variance, okay? The mean is not affected. Whereas selection, so we have an example here where on the left-hand side, it's no selection and small population on the top, a large population on the bottom. And you can see that for selection, it affects both the mean because the trajectory becomes more deterministic, going to fixation in the case of positive selection. And it also reduces the variance in the allele frequency trajectories. Okay. So in principle, if we knew the trajectory, we should be able to tell something about the parameters that lead to that, N and S, so these two parameters. So the way we model the data to be able to infer those parameters is to use what's called hidden Markov models. This is something that's been proposed a while ago by Bolbach et al. So the idea is that you allele frequency trajectories, the x's, they are unobserved variables. But if I knew those variables, if I knew the allele frequency trajectory, then it would be easy to calculate the likelihood of my data. It's just a binomial. I'm just drawing some alleles from this trajectory, from this allele frequency. Okay. Okay, so of course I don't know those, but in a hidden Markov model, what I have to be able to do is I have to be able to explain what is the allele frequency at time t, or k here, sorry, given that I was at what was the allele frequency at the previous generation. Okay. And if I can do that, then I have my transition probabilities, the Q's, and the emission probabilities, the G's. And if I can compute both, then I can do whatever I want. So I just have to be able to explain what's going on. So for the G's, I would say it's just binomial, just sampling from, it's easy. I don't have, problem solved. I guess if you do pool seek, you can include the pool size in there to have some random effects of pool size, but that's just experimental design. Now for the Q, it's a bit more complicated. So to think about the Q, if you think about a population parmixia with non-overlapping generation, constant population size, what happens when you go from one generation to another is that you're just sampling the new alleles from the previous one. So it's a binomial where you're taking big N and the probability is just the xt, the frequency of the previous generation. That's just drift. And you can do that, and that goes forward. Okay. Now, if you have selection, it means that some of the genotypes have more chance to reproduce than the others, and that depends on this selection coefficient S, which is the classical selection coefficient used in the PopGen literature. And in this case, then your expected, well, the law of your frequency at t+1 given xt is a binomial too, but now it's a fitness function of xt. So it's a bit more complicated. It depends on S. And the problem is that because you're not sampling every generation, you need to explain what is the distribution of x after u generation, say. So it's just this matrix P that you raise to the power u. So under a Wright-Fisher model, you can write these transitions. And so you could say, okay, so everything's fine, I can write the transition. Well, the problem is that this matrix Q has dimension the size of the population times the size of the population. So it's a very big matrix potentially. And when you raise that to the power U, so the matrix P also, it's very expensive computationally. And also you have a lot of memory load because you store those big matrices. So the alternative to using this is to find good approximations. So in the Bollback paper I was mentioning before, they were using the diffusion approximation. And in our case, we used a parametric distribution. So we did that with Simon and Cyril Paris, who was a PhD student with us, which is a distribution that is computationally tractable and it's kind of accurate. And it's better than the diffusion approximation in our context where we have small population size and strong selection. So it just allows to compute those transition matrices much faster than you would in with the Wright-Fisher model. Okay, so what we did recently, and with Paul and another student, Mathieu, who was working with us, is that we implemented all this in a Python package called selAnytime. And what it does is that it takes and inputs the matrix of allele counts, reference and alternative, at all the loci in your genome, and then it estimates. So it estimates any, assuming there is no selection, and then it estimates S conditioning on this effective population size. And not what we are doing with Paul is that we're working on jointly inferring any and S and not assuming there is no selection. Because when you do assume there is no selection and you estimate any, you're actually biasing your estimate of effective population size. So if you're interested in this, there is a paper just came out. There was a preprint for a while. Now the paper came out in Peer Community Journal. And there's a Python package that you can try if you want. And this is an example. This is just to show what happens when you use a package to estimate any. For different any, you get the likelihood, and you can just pick your best one. And the red curve is the parametric approximation using the beta with spikes distribution. And the blue curve is the right Fisher, and you can see the right Fisher, you can only compute it up to, say, 1,000 population size. Afterwards, it becomes computationally intractable. Okay, so with this package, we can estimate a selection coefficient for every locus on the genome, if you have genetic time series. And because we compute likelihood functions, we can also estimate standard errors around this parameter. Okay, so we get, and that's what you need to remember for the next part of the talk. Okay, so now we're going to be using this to infer polygenic response to artificial selection. And so the first part of this slide is what I said before. And what I want to talk about first is how we design some simulation to fit artificial selection experiments we have. And what we wanted to look at first is the joint distribution of the effect of markers on the trait, on their selection, and their selection coefficient. So what does it look like when I select for a trait in a population, and I estimate the marker effects on the trait using a GWAS, and I estimate the selection coefficient from allele frequency trajectories? How do these two quantities, beta and S, look like when I look at them together? So our context, because this is real data that we will analyze, but later, so you will not see, is this artificial selection experiment that was conducted by some colleagues. So they crossed some pigs and then selected in two divergent lines for one trait, which was efficient residual feed intake, which is a measure of feed efficiency in the pigs. Doesn't matter for today, but what happens at each generation is there are 6 males are selected and they're crossed to 42 females. And the crosses are not random. They are made so that there is not too much inbreeding going on. And so you can see the sample size are pretty small, right? The population size are pretty small. Our problem here is that we want to be able to understand the genetic architecture of the trait from these experiments and then try to predict those traits in another population that is distantly related. Okay. So all the results I will show about the simulation is reproducing this selection scheme based on that. And yeah, and there are two divergent. Okay, so when we did the simulations, the problem is that we're interested in the genetic architecture and how it evolves. But when you want to, if you want to simulate a quantitative traits, we didn't want to condition on what was the starting genetic architecture, because we believe that if you change that, that could change maybe a lot, and there's a lot of stochasticity. So what we did is that we decided to simulate an infinite number of QTLs plus 1. So what we did is we simulated just one QTL, that's your X here, and its effect beta. And then we also simulate a polygenic effect that is a continuous trait. Okay, so it's like you have an infinite number of QTLs that contribute to your polygenic effect, and then you have one more QTL, which is the one we're going to look at. Okay. So your total genetic effect is the QTL plus the polygenic component, and it's actually something that was done in this paper a few years back. Okay, so so this is just a static view. So what happens when you transmit this to your offspring? So for the QTL, it's easy, it's just Mendelian inheritance, right? So for the polygenic effect, what you simulate the new value of a new individual, you take the mid parent and then you add some meiosis random, right? So it's just the meiosis deviation is driven from a normal with the additive genetic variance divided by 2. But the thing is that we also want to account for the fact that because of inbreeding, you have a decreasing genetic variance over time. And so for that, we account for that using this technique described in this paper. So we can transmit a quantitative, a polygenic value this way. And so we don't have to simulate all the other QTLs on the genome, just say there's an infinity of them. And to do these simulations, we use SimuPop, which is, I guess, an old software for population simulation. Slim is more trendy today, but that works too. Okay. So we simulated, so we took this parameters value from the true experiment. So the trait in question is a heritability of 0.1. And we started with a QTL that was at 0.5 because that's where response to selection is the biggest. And then we also simulated neutral markers. So they are neutral, they're unlinked. There is 1,000 chromosomes, if you want. And we wanted to be in the simplest case. And you'll see things are not that simple, even in this simple situation. Okay. So when you look at what happens, everything's fine. So so there's one line that goes up, one line that goes down. Whatever is the true effect of the QTL, the trajectory of the response to selection is the same because the genetic effect, the overall genetic effect is the same. Right? It's just that some of it comes from the QTL, some of it comes from the polygenic component, and depending on the effect of the QTL, you will see in a moment, the contribution of both is not the same, but the overall trend of genetic value is constant, whatever is the QTL effect. Same thing, you can see how the genetic variance decreases through time because of selection and inbreeding. But that doesn't depend on the QTL effect. Okay, so our simulations in terms of response to selection don't depend on the QTL effect for this. However, if you look at so this is like the average trajectory of the allele frequencies through time. And of course, this depends on the QTL effect. So when the QTL is strong, it will respond to selection fast. And when the QTL effect is small, like it's gray area here, it behaves essentially like a neutral marker. And yeah, a neutral markers, it just randomly drifts. And you can see the two lines, so the mean and the max lines, the plus and minus line. Of course, the QTL evolves by diverging in these two lines. Okay, so we have these simulations, and now we want to, so we estimate quantitative effects, and we estimate selection coefficient. So one thing I want to stress out is that for the GWAS we do for the beta, we are actually using the actual generative model. So everything should be fine, right? We are estimating beta under the model that was used to generate the data. And actually, when you look at the QTL, you have a very big, nice correlation between beta hat and beta. And for S hat, we use this package I was talking about. And here there is no true S, right? Because we don't know what S is. S is just a consequence of phenotypic selection on the trait and on the beta. That's really the hard part. We don't know what is the selection coefficient of a locus when you select on a phenotype because it's really indirect. There are some theoretical work, especially done here, on certain very specific situations where you have like stabilizing selection, large population size, and stuff like that. But when you have small population size, strong selection, we don't know what the relationship between beta and S should be. And mathematically, I would not even know how to start. So that's why we do the simulation. Okay. So what I will be showing is, so this is a relationship between the delta in the selection, so the difference in the selection coefficient estimated between the two lines. It's just easier. So this is delta S hat using selenotype, and the beta hat that we get from the GWAS at the QTL. So when the QTL is strong, you have a very strong difference in selection coefficient between the two lines because the selection coefficient is very positive in the plus line, very negative in the minus line. That's what happens. So that's good. There's a good correlation between the two effects. The problem is that when we looked at neutral markers, so individually, the two parameters are not biased, right? If you look at the marginal distribution of the delta S, it's around 0, nothing wrong. The beta is not biased, around 0, some variance. But there is a correlation between the two parameters. Okay. So it means that the marker effects of loci that are randomly differentiating between population just due to drift and polygenic selection tend to have higher estimates of their effects on the trait. Okay, and this is not, well, we didn't expect that. We thought there would be no correlation between S and beta when you have, but apparently there is, and it's substantial. And despite the fact that we're actually fitting the correct statistical model, the one that we use to generate the data. And actually, this theoretical result, I mean, this result that we can produce via simulation is something that has been observed empirically in the literature. This is something that is related to the problem of false signal or false, like debatable signal of polygenic adaptations in humans. So you had these studies, and there's this very nice paper by Berg and collaborators in 2019 in eLife, and also good discussion in November and Barton in 2018. The fact that so you could think of selection acting on neutral markers unlinked as some kind of subtle population structure. And this subtle population structure actually creates these biases in the beta estimates. And that's why for a long time, there was these papers about how there is selection for height in Europeans. And then when you actually use betas that are not affected by population structure, this signal almost disappear. In another context, there's something that happens in animal breeding, what's called erosion of breeding values. The basic idea is that, so what happens in genomic selection is that when a new calf is born, you genotype it, and you estimate its breeding value to know if it's going to be good for whatever trait you're looking at. And what's been shown is that your prediction of its merit is usually the good ones are overestimated. When they have daughters, say for dairy bulls, later you find that their genetic merit is not as good as you would have expected. And that's because they tend to carry alleles that increase in frequency in the population, and there are biased effects upward. And this is also, there's also a colleague who published a paper recently about this question, and I think it goes in the same direction. So what it means that when you use these standard methods, and in situations, very simple situations, so imagine if you have linkage disequilibrium, and that becomes more complicated. There is a bias in your inference of genetic architecture on the trait. Okay, but that's taking things independently, right? Or only using beta hat as a. But we're saying, okay, but now with our simulations, we see this correlation, so maybe we can correct for that and we can improve on that. So that's what we've done after that. Oh yeah, I wanted to show you sorry, before going to how we try to correct for this, what's the consequences in terms of predicting the values of traits. So on the left-hand side, you have genomic prediction. I'm trying to predict the genetic value, but I'm in the simulation where there is no QTR. So none of the markers in the data are causal. Their effect is all 0. And yet, within your population, you can predict the trait, because you predict the kinship of the individuals, and so you have good prediction performance. But there is nothing that's actually linked in your marker. I mean, that's causal in your trait. However, if you train your prediction model on this, and then you try on simulations where there is a true QTR, and you try to predict the genetic values of individuals from another population, from the other line. So you're fitting your model in the minus line, and you're trying to predict the plus line. It doesn't work at all, because the animals are too distant, and your effects are biased, and so your prediction model is just bad. And what we want is exactly the opposite. We want no prediction on the left and good prediction on the right. So can we do that? So what we have done is we said, okay, we can combine. So the idea is that if I see a locus that has high selection coefficient and also high beta, maybe I can correct my beta for taking account this. So the principle of the method is to take our individual analysis at any time GWAS and combine them together with the meta-analysis method to integrate our uncertainty in their values in this distribution and get some idea of what is the posterior distribution. So get new prediction for beta and S that combines both approaches. And to do that, we've tried different things. And the one that ended up working well for us was using this method called MASH, proposed by Urbut from Matthew Stevens' group, which is a kind of a generic method when you have multiple conditions and you have estimates of one parameter for multiple conditions, and you want to combine things together. And it's a method that's based on summary statistic, which is very nice because you can do whatever. So you can do your GWAS on one side, your selection coefficient estimation on the other side, and then you just bring your estimators in this method and you get the posterior for your parameters of interest. Okay. I won't go into how this method works. Just want to mention one thing. So it's modeling the joint distribution of beta, the selection coefficient in the plus line, selection coefficient in the minus line, using a mixture of normal distributions. And the important thing is that there is a possibility to give what is the expected covariance under neutrality. For a neutral marker, what is the expected covariance between your parameters? And this is what we can do with simulation. So the idea of the method is that I can do simulations, and in real data, I could do simulations. I could just do gene dropping in my pedigree, and I can look at what is S, what is beta. So I can derive what this looks like when there is nothing going on. And I can put that in the method. So I can correct for this bias to get new estimates of beta, S minus and S plus. And when we do that, we still have good correlation between the new delta S. So this is a delta S before, but the new beta tilde. We still have a very good correlations between the two. So we don't lose a lot of power at the QTL. We're still having a good estimation of the QTL effect when there is an effect. However, now there is almost no correlation between delta S and beta tilde. So we removed this correlation that was spurious and that was hurting us. So when we try to predict genetic values with these new betas, within a population, we don't have any prediction power anymore. We cannot predict the trait with markers that are just not relevant for the trait, which is good. However, when we're trying to predict the plus line, having trained the model on the minus line, we do have predictive power as far as we can have. We can only predict the QTL. Of course, we cannot predict the polygenic component because the other QTLs are not in the data. So using this approach, by combining population genetic information and quantitative genetics information, we can actually restore predictive power when we have no power when we shouldn't have any. Yeah. So two conclusions. So we see that influence of the genetic architecture of a trait can be highly biased when you are doing analysis of a population that is under strong selection. And I would say, which is our case, right? Because this is typically the population we're working on. So, and considering both selective effects and quantitative effects can actually improve inference. And in this situation, I showed you quite a lot. So that should allow, and that's what we're going to try to do now, and using real data to test it in real life situations, we should have better prediction in the marker effects, and so better transferability of inference to new populations, which is a very big topic in animal genetics, especially when you want to be maybe able to use more genetic diversity. You want to be able to work with more populations. And in a more maybe fundamental problem, maybe it can help understand better the evolution of complex trait under strong selective pressures, because you have a better view of what is the actual genetic architecture of your clone. Yes, and to finish, I want to thank the people who have been working with me on this, or I've been working with them, both ways. So the FitSec project with this PIG project I was mentioning, my colleague Julie Demar and Hélène Gilbert. For the polygenic adaptation, the second part of the talk, that's really the work of Elise. And the genetic time series, so this Pelani time, there was Mathieu Hull, who was a student working with us for a while, and then Paul now is working with us is Simon Boitard, another colleague, Miguel de Navasquez. They're both in CBGP in Montpellier, a lab where they also do Drosophila experimental evolution. And yeah, and thank you for inviting me and for your attention.